In today's episode, we'll be going through enzyme exam questions. The way I'm going to do this is that I'm going to show you from start to finish how I'm thinking, approaching, and answering the questions. And remember that the mark scheme is written in the description box below. So let's begin. So the question says, why is pH not a controlled variable in this investigation? Now what I would do is think, well, what is a controlled variable? Is one that you do not change and it stays the same throughout the experiment. We can see that this experiment is about the effect of temperature, right? So that's the thing you're changing and thus would be the independent variable. So at this point, what I would do is identify what the control, independent and dependent variable is, because that's probably going to be the answer to the question. So now I'm going to look at where pH is mentioned in the box above, and that's in the third bullet point. Now because it talks about 8.3 and then above and below it, I can assume that the pH would be the dependent variable because that's the thing you're measuring. Now the next question says, identify one variable that has been controlled in this procedure. So we're going to read the box above. It talks about the volume of the alkaline solution. It talks about the temperature of the water bath, the volume and the concentration of the lipase solution. And it also talks about the time, so for 5 minutes at 30-second intervals. That in itself is like 5 marks, but of course there's a 1 marker, so we can just write any one of those points. Identify one variable other than the pH that has not been controlled in the procedure. So look at the box and think, what has not really been specified? I can see that a few drops of an indicator was added. How many drops? Maybe you would put 8, but I would put 2. So that's something we can write about. The procedure required the solution to be stirred and then drops of solution to be placed on a white spotting tile. Suggest why this procedure was followed rather than simply adding indicator to the test tube, stirring the solution and looking for the colour change in the test tube. Think about it logically, and to be honest, this practical was also done in GCSEs. If you put something on top of a white tile, that's gonna produce like contrast. You get to see the colour change much more easily. So that's why sometimes you can actually use logic for your answers. What can be concluded from the results in table 4.1 about the optimum temperature for lipase activity? To identify the optimum temperature, we need to see where the time where the solution did not remain pink stayed the same, and we can see that's between 30 and 35. But of course we can't write it's 31 or 32 or something like that because we aren't given those values, those intermediate values, so we can just say a range of 30 to 35. Describe 2 different ways in which the procedure could be modified to obtain a more accurate value for the optimum temperature for lipase activity. I gave it away in the last question about saying, oh, you need more intermediate values between 30 and 35 degrees Celsius. So at least that's 1 point. The reason being is because we actually don't know what the exact temperature, the optimum, happens at. So that's why. Let's have a look at the box. Another thing we can say is about time. It says at every 30-second interval. Well, let's do every 15-second intervals. So we need to take samples at a more frequent time interval. That's 1 point. Another point one can say is about the use of a pH meter. That's quite often on the mark schemes, and the reason for that is to obtain a numerical value opposed to just relying on subjectivity of, oh, well, this colour is going to be this at that pH sort of thing. Okay, and that's the end of the video. Remember, the 2 most important exam techniques are read the question first and state all the obvious points, because I can guarantee you're going to get the marks from that. Over to you. Try some questions and let me know how it goes.